Hi, my name is Andrew Liang, and today I'll be presenting on the identification of SARS-CoV-2 main protease inhibitors utilizing Acceptor Warheads. This research is part of a larger international effort towards drug development against the SARS-CoV-2 virus. As of January 20th, 2021, there have been over 110 million cases of SARS-CoV-2, resulting in over 2.4 million deaths worldwide. SARS-CoV-2 virus is a type of beta coronavirus. It has up to 82% sequence similarity with other beta coronaviruses such as the 2012, 2002 SARS, and 2012 MERS. Now, recent developments in SARS-CoV-2 treatments such as the Pfizer and Moderna vaccines show promise, but issues remain with mutations and future strains of beta coronaviruses. Thus, there is an imminent need for novel broad-spectrum antivirals that can target all beta coronavirus classes. SARS-CoV-2 enters human cells through a receptor known as ACE2. Upon cell entry, the virus begins replication, which requires the assistance of many proteins. One of the most important is the main protease. The SARS-CoV-2 main protease, also known as Mpro, facilitates the activation of viral proteins necessary for RNA replication. Its key role in viral pathogenesis makes it a desirable target for drug development. In addition, there are no enzymes similar to the Mpro in the human body, minimizing potential inhibitor side effects. Furthermore, protease inhibitors are viable antivirals, as demonstrated by the FDA-approved drugs lopinavir and tapronavir, which target the HIV-1 protease. Together, this makes the Mpro a viable drug target. To target the Mpro, I first thought to understand its structure, which is shown above. Here I'd like to point out two important things. First, this enzyme is a protease. These are protein-cutting enzymes, and as an enzyme, it restores itself after hydrolyzing a substrate, so it can be reused. Second, this is a cysteine protease, which means that the part of the enzyme engaging in catalysis is a thiol. Knowing this, our inhibitor approach focuses on inhibiting a thiol group by preventing the restoration of the enzyme. Thus, seeking irreversible inhibition of a cysteine protease, we introduce the star of the show, which are mycoreceptors. Mycoreceptors are a class of compounds containing an alpha-beta unsaturated carbonyl, which acts as the main trap to covalently inhibit a thiol, such as the main protease. Furthermore, these compounds have electron withdrawing groups, which are shorthanded EWGs to either side, or both, to reduce electron density around the carbonyl, making it a better target while preventing enzyme reactivation after being inhibited by the mycoreceptor. Now, modifications to these EWGs at either side can produce large numbers of different mycoreceptors with varied efficacy against the Mpro. Thus, the problem statement in the status quo, there exist limited coronavirus targeting mycoreceptors, of which none are sufficiently potent to justify in vivo studies. And thus the research question, can rational basis design be applied to produce large numbers of mycoreceptor-based main protease inhibitor candidates? I hypothesize yes. And second, from our library of Mpro inhibitor candidates, can we identify viable compounds with superior binding affinity to the SARS-CoV-2 main Mpro when compared against existing mycoreceptor inhibitors? Once again, I hypothesize yes. This is an independent study I conducted, developed, and designed. I worked with 56 compounds over the course of 30 plus weeks. First, materials and methods. We start our research methods with the rational design of mycoreceptor inhibitors. This design requires three key improvements from its basic structure, starting with EWG modifications. An existing mycoreceptor is the α-ketoamide. Now we will be using this as the control. Now there are a variety of alternative EWGs, and recent studies suggest that sulfonate ester EWGs may perform better than these control α-ketoamides. Thus we propose the following classes of EWG modified mycoreceptors. We have combinatorially created 7 EWG classes of mycoreceptors. At the R1 position, which is shown over here, there are modifications between ketones and amides. At the R2 position, there are modifications between amides, esters, sulfonamides, and sulfonate esters. Class 1 compounds, which I'm currently circling, are the control. These are the α-ketoamides. Now, our second key improvement is based on amino acid substitutions. I want to mimic the Mpro's natural substrate to enhance the Mpro's recognition of our compound inhibitor. We know that the Mpro has a high binding affinity for proteins because its job is to lyse proteins. Thus, I decided to attach amino acid side chains to either side of the compound as a second optimization. This helps mimic the structure of a protein, so the compound and inhibitor is more likely to attract the main protease. In 2020, Sun and others reported that the binding pocket of the Mpro is anionic. Leads us to use something cationic. Here we propose 8 amino acid pairings, all of which contain the cationic amino acid lysine or arginine. Now our last improvement is based on pharmacokinetics. I've attached protecting groups to all unwantedly reactive parts of the compound, as shown in purple here. In combination with EWG modifications and amino acid pairing modifications, we have assembled our novel mycoacceptors. Thus, in summary, using 7 different EWGs and 8 different amino acid pairings, we have a total of 56 compounds to investigate. Now, given these 56 compounds, we ask the question, are there any Mpro inhibitors with binding affinity higher than the control among these novel compounds? I will be using in silico modeling and molecular docking to measure the binding affinities of all mycoacceptors. Then I'll use statistical analysis to compare docking results across the classes. With these 56 compounds designed, I draw up their structure in silico using the tool Avogadro. Then I perform DFT calculations to predict their structure in a water solvent. This allows me to predict the structure of these compounds within a human system, and as structure dictates function, this is a very important step to make sure that we can correctly predict the function of these compounds. Lastly, I will utilize molecular docking to determine interactions between the SARS-CoV-2 Mpro and my inhibitors, finding delta G of binding in an optimized binding pose. Delta G of binding is a measurement of the energy released during the binding process. A negative delta G means energy is released. More energy released means that the reaction is more easily going to happen. With the molecular docking results, we begin a statistical investigation of 4 main questions. First, do EWG modifications affect mycoacceptor binding affinity? Second, which EWG modified mycoacceptors have higher binding affinity than the control? Third, do amino acid side chain modifications affect mycoacceptor binding affinity? And lastly, which amino acid side chains have higher binding affinities? First, I created summary statistics and box plots by EWG and amino acid classes. I will then do a 2-way ANOVA test for the differences of mean affinities across EWG classes and amino acid groups with a p-value of less than 0.05. The null hypothesis is that among EWG classes 1 to 7, all of the binding affinities will be consistent. And the other null hypothesis is that among amino acid pairings 1 through 8, the binding affinities will also be consistent. The alternative hypothesis, on the other hand, is that the mean binding affinities of different EWG classes are not all equal, or that the mean binding affinities of different amino acid groups are not all equal. So if the null hypothesis is rejected, multiple comparisons will be conducted using Taki-Kremer tests to identify which EWG or amino acid pairing was statistically different. All the statistical analysis done in this research uses SAS 9.3. Next, let's take a look at the results. So we start with a visualization of the molecular docking results. This is the 3D structure showing the interactions between the SARS-CoV-2 main protease and the control mycoacceptor. The white complexes are parts of the main protease, and the blue/teal color is the inhibitor as shown on the screen. So these are the raw data binding affinity results for each individual mycoacceptor measured in delta G of binding. They've been colored using a key, with warmer colors demonstrating stronger binding. I've listed the columns by 7 EWG classes and the rows by 8 amino acid pairings. Class 1 compounds are the control. Visually, columns 2, 4, 5, 6, and 7 EWGs appear to have more warmer colors. The same goes with the amino acid group RR. Now these are the summary statistics and subsequent box plot analyses of the 7 EWG classes. On the y-axis is the binding affinity in kilocalories per mole. A lower or more negative binding affinity means stronger binding and thus stronger inhibition. By looking at the box plot, we observe that classes 2, 5, 6, and 7 appear to have stronger binding affinity when compared with the control group. Furthermore, these are the summary statistics and box plot analyses of the 8 amino acid pairings. By looking at the box plot and means, we observe that the RR pairing has the lowest mean of negative 10.6, and furthermore from the box plot, it appears to have the strongest binding affinity as well. Now here are the results for the Anova Analysis and Taki multiple parameter comparisons for the 7 EWG classes. The p-value from Anova Analysis shows that the binding affinities are significantly different across these different classes. The last column shows the Taki-Kremer groupings, which I'll delve into right now. So here is a model least square means and 95% confidence interval of the Taki-Kremer comparisons. EWG classes grouped with a different letter are significantly different from each other. Thus, we see that classes 5, 2, 6, and 7 are highly significantly different from the control, while class 4 is significantly higher than the control. Now we move on to the Anova Analysis and Taki multiple comparison results for the 8 amino acid pairings. The p-value once again shows that the binding affinities are significantly different between the 8 amino acid pairings. Here again is a model least square means and 95% confidence intervals for the Taki-Kremer groupings for amino acid modifications. We see that there are no significant differences among these 7 pairings, but the RR amino acid combination is highly significantly different from almost all the pairings. Specifically, it is highly significantly different from the pairings KE, KR, and KS. It is also significantly higher than the RE and RY pairing. Thus, the conclusion: both EWG and amino acid modifications have a significant impact on mycoreceptor binding affinity to the main protease. Specifically, R1 amide-containing and R2 sulfone-containing compounds, which would be classes 2, 5, 6, and 7, have significantly higher binding effects than the control. Furthermore, mycoreceptors containing arginine in both A1 and A2, so these are the RR compounds, have significantly higher binding affinity than the others, making it the optimal construction. So moving on to the study strengths. This study was based on principles of rational basis drug design. Specifically, we designed our drugs based on an analysis of viral pathogenesis, inhibitor structure-activity relationships, and drug receptor binding mechanisms. Furthermore, we had strategic use of optimal chemical moieties identified by existing research, such as Reddick and Zhang. This means that our research is more efficient than random high-throughput screening, which screens hundreds of thousands of different compounds without justification. Moving on, we utilize molecular docking in high-throughput screening. Molecular docking is a powerful tool in modern drug discovery that is highly efficient and very simple. It dramatically shortens the development period of novel drugs and results in cost saving as compared to the synthesis of all the different compounds and subsequent in vivo testing. Together, this allows for the rapid and fruitful cutting-edge research on SARS-CoV-2 inhibition. Furthermore, this test is extraordinarily accurate as molecular docking has been designed and optimized for decades now. Lastly, I utilize statistical analysis, specifically two-way ANOVA and Tuckey-Kramer multiple comparisons. In summary, this research explored novel SARS-CoV-2 inhibitors to tackle the global health crisis, identifying the following 3 types of mycoreceptors for further investigation in vivo and in vitro analysis. Thus, I overcame current limitations to mycoreceptor inhibitors by identifying key optimizations to heighten their potency against viral proteases. I developed a feasible and efficient approach for the identification of SARS-CoV-2 main protease inhibitors. Given a high amino acid sequence similarity across the mycoreceptor and the main protease of beta coronaviruses, this approach and the selective inhibitors identified in this research can be applied to other beta coronaviruses, with potential for broad-spectrum antivirals. Now, due to time constraints, I've tested 7 EWG classes. In future work, I would like to expand the study on related EWGs to confirm the identified trends. Also, I would investigate structure-activity relationships responsible for the identified patterns, providing guidance for future rational drug design. Lastly, I'd extend this research to other pathogenic beta coronaviruses. I would like to acknowledge my mentor, Dr. Gulin Rather, for his guidance and inspiration throughout this project. Furthermore, I would like to thank Mr. Edward New, Dr. Alden Blair for their advice, my classmates at school for their feedback, and my friends and family for their constant support throughout this tough pandemic period. Here are all my references. Thank you so much for your time, and I'm open to any questions.